Megan, can you tell me if we're live? Okay, we're going live now. Good afternoon. Wonderful to see everybody. I would like to introduce our speaker for today, the first one in the series of primate conversations for this term, Enquye Negash, talking to us about woody cover and hominin environments. Enquye is from Ethiopia. She obtained her Bachelor of Science degree at the University of Addis Ababa in 2008. I believe 2008 is when I first met Enquye in Addis Ababa, doing work at the National Museum of Ethiopia there. And I think at that time, Enquye was finishing her Bachelor of Science with an interest in paleontology and was looking for opportunities to do further work in the museum. And I think that started a long collaboration with Enquye in topics that we have in common related to hominin paleoecology and hominin evolution. Enquye then obtained her Master of Science, also from Addis Ababa University, 2012, I believe. And then she joined me in Washington at the George Washington University in the Hominin Paleobiology program, where she obtained her PhD, finished relatively recently, a couple of years ago, doing work that, in my view, is truly cutting edge. I hope that you get a sense of that today, where she takes data that she has collected from many parts of Africa, from Ethiopia down through the Rift Valley, all the way south to Mozambique. Enquye has worked in Gorongosa with us, where we have an ongoing project, as well as extensively in Ethiopia and the Great Lakes region of Africa. She has a postdoc now at George Washington University, and she has been publishing some truly impressive papers, including the most recent one in PNAS, where she analyzed a very large data set of fossil mammals, looking at stable isotopes and looking at the main dietary trends across all kinds of lineages of mammals in the Pliocene and early Pleistocene of the Omo region of southern Ethiopia. So the work today, I think, is one of critical importance for us to understand the kinds of environments in which hominins evolved to help us also understand how our key adaptations may have come about through evolutionary processes. But it's a real pleasure to have Enquye with us, and I leave you with her. Thank you, Enquye.
Thank you very much for inviting me, Renee. I'm very happy to be here. And yeah, today I'll be talking to you about woody cover and hominin environments. So the hominin fossil record indicates that the earliest members of our lineage appeared around 7 million years ago, and that's in the late Miocene. In this very sparse fossil evidence is relatively better represented in the Pliocene and Pleistocene, where we have this multiple hominin lineages, including gracile hominins and what we call robust hominins, but also including the genus Homo. And throughout the evolutionary history of our lineage, morphological as well as behavioral evolution has occurred, and several hypotheses have been proposed to explain these adaptations. And most of these hypotheses are in one way or the other related to environmental change. And as pointed out by multiple previous studies, although it's very difficult to assign this cause and effect relationship between this environmental change and hominin evolution, we see that major trends in human evolution or major events in human evolution are associated with global as well as regional environmental change inferred from the continental, but as well as the marine record. And a few examples are presented here. As you can see in this figure, we see the occurrence of sapropel deposits that indicate alternating wet and dry cycles. From the continental record, we also have record that shows expansion of C4 grassland ecosystems from soil carbonates, from plant wax biomarkers, but also there is evidence from the fossil faunal assemblage that indicates an increase in the number of grazing species. And based on this different environmental records, different hypotheses about hominin evolution have been proposed. And a way of testing this hypothesis is by studying the paleo environment and how it changed through time from this fossil bearing sediments in Africa. And one approach of doing that is reconstructing or inferring about the vegetation on the landscape and assessing how the paleo landscape changed through time. And there are several ways of doing that, or there are several approaches to reconstruct vegetation. We can do that by looking at the plant fossil record, including, let's say, macro plant fossils such as fossil wood, leaves, and seeds. We can do that by looking at plant microfossils such as pollen and phytoliths. We can also do that using more or less indirect techniques, such as looking at the fossil herbivore assemblage, its taxonomic composition, ecomorphology, or looking at the diet of herbivores. But again, we can also get at reconstructing vegetation by using the sedimentary record, such as paleosols and proxies within paleosols. And my research, as Renee mentioned, employs using different vegetation proxies. And for this talk today, I'll be presenting to you about my work that employs using proxies that are in the soil in both modern and fossil soils, but also using the diet of herbivores to infer vegetation change in the Pliocene and Pleistocene from fossil-bearing sites in Eastern Africa. So one approach of studying vegetation structure is by studying woody vegetation cover. And by woody cover, this simply refers to the amount and the distribution of woody vegetation on the landscape. And a very simple way of defining it is, if you imagine lying down on the ground, you can think of it as how much of the sky you see. Woody vegetation cover has many determinants, and the amount and also the distribution in modern ecosystems is determined by complex interaction between resource availability, such as water and nutrients in the soil, biotic interaction between plants, such as the competition between trees and grasses, but also other extrinsic factors, such as disturbance by the action of herbivores, by the action of natural fire, but also by the action of humans. And several studies that looked at woody cover at a continental scale have shown that precipitation is among the major determinants of woody cover. But in addition to that, we know that factors like soil texture, which ultimately determine how water flows through the soil, how water is retained through the soil, and also how well plants grow on the soil, are also key determinants in addition to precipitation. Therefore, due to this geographic differences in these different determinants, and by that I mean differences such as this, the biotic composition, the herbivore composition, and climatic conditions, but also the geology that ultimately determines the soil type. Because these conditions vary from place to place, the amount of woody vegetation cover also varies from place to place. And if you ask why studying woody vegetation cover is important for mammals, in general, it's because, again, the amount and also distribution of woody vegetation is a key factor that determines primary productivity, species composition, species abundance, distribution, but also the overall heterogeneity of a landscape. And for example, as shown here, if we take primates, for example, like chimpanzees, they usually occur in forests, while other herbivores like a wildebeest would prefer more open grasslands. Sorry. And in terms of this ecosystem services provided by woody vegetation, again, its distribution and amount on the landscape is going to dictate where species can access their dietary resources, where they can find shelter, shade, tools, but also where they can find a niche space and a place where they are protected from predators. So when it comes to hominins, just like any other mammalian species, hominins likely benefited from this resources and ecosystem services provided by woody vegetation. But in addition to that, woody cover is particularly important to hominins because it's related to key adaptations in our lineage, such as our bipedal walking, endurance running, tool use, as well as thermoregulation and shade-seeking behavior. Therefore, to be able to better understand these adaptive strategies, we need to have a clear understanding of the woody vegetation cover that was on the landscape, so that we get at this key adaptations in hominins. But before making this reconstructions and before making this woody cover reconstructions in the past, first we need to have a clear understanding of the amount and distribution of woody vegetation in modern ecosystems. So therefore, a major component of my study is focused on conducting a high spatial resolution quantitative study of woody cover in modern African ecosystems. And that's mainly using national parks or reserves, but also in terms of proxies, using proxies that can potentially be inferred or preserved in the fossil record. And again, the main aim of focusing on modern ecosystems is to be able to provide a more refined paleolandscape reconstructions or vegetation reconstructions for hominins. And to do that, I studied woody vegetation cover in parks that are located in Ethiopia, Rwanda, and Mozambique. And this work consisted of studying woody cover in 9 different national parks. And these different parks were selected to cover a wide range of climatic as well as woody cover gradients, from relatively arid and shrub-dominated landscapes, as we see in the Awash, to very dense forests that we see in Enquye. But in most cases, each park has a wide range of woody cover types. And here I have a couple of pictures just to show you the different landscapes in this national park. Here you can see Afro-montane forests in Volcanoes National Park in Rwanda. Again, here we have forests in Bali Mountains National Park, but you can also see this shrub and bushland dominated landscape in the Mago. We have woodland dominated landscape in Gorongosa, and we have bushland and grassland dominated landscapes in Akagera and Nechisar National Parks. And to apply what we learned from these modern ecosystems to the fossil record, my work is focused on reconstructing vegetation from hominin bearing sites in the Omoterkana Basin, which is a site located in southern Ethiopia and northern Kenya. And there are multiple formations, geologic formations in this basin. We have the Mursi, the Osno, Shangora, Dachukwi, and Kubifora. But my work focuses on the Shangora, which is found on the Ethiopian side of the basin. And just to give you a little bit of background, the Shangra Formation is one of the most prolific hominin-bearing sites in Eastern Africa. It comprises of about 800-meter thickness composite of fluvial, lacustrine, and deltaic sediments. This formation is very unique as it represents a relatively long and continuous record, which is also a strata that is underlain by volcanic ash, which allows us to have radiometric dates for the different members. And we have about 12 different members in this formation. Again, each layer is commenced by volcanic ash layer. So you can see here we have the ages for the different members. And more than 50,000 fossil specimens have been collected from this site since the 1960s. And in terms of the hominin fossil record, 4 hominin taxa are known from the sites, including the genus Australopithecus, Paranthropus boisei, Paranthropus ethiopicus, as well as the genus Homo. And detailed paleoenvironmental studies, again, have been conducted in the sites using multiple proxies. But what I'm going to present to you is looking at the geochemical record from this site. And for my specific study in both modern and fossil ecosystems, I use stable carbon isotopes. And the use of carbon isotopes is based on the understanding that C3 and C4 plants, C3 plants being trees and shrubs, and C4 plants being grasses, they follow different photosynthetic pathways, which results in a distinct carbon isotopic composition. So when we're dealing with modern soil, the organic carbon in the soil or in surface soil specifically is going to be derived from the plant biomass that's above the ground. So its isotopic composition is going to provide us with an integrated measure of bulk stable carbon isotopes of the overlying vegetation. But when we're dealing with fossil-bearing deposits or fossil-bearing sediments, we will use pedogenic carbonates, which form at depths within the soil profile and form in isotopic equilibrium with soil CO2, which simply means that they're going to record the vegetation signal with a consistent fractionation factor or a consistent offset from the vegetation. Similarly, when we're using the diet of herbivores to infer about the vegetation, Plants are, again, the primary source of carbon for herbivores, and the carbon isotopic composition of the food that they consume is going to be recorded in the body tissue of herbivores. So again, tooth enamel is going to record the carbon isotopic composition of the food with a consistent fractionation factor or consistent offset. Therefore, by studying either modern soil, pedogenic carbonates, or the diet of herbivores, we can talk about the vegetation. So now coming back to the woody cover concepts, in order to quantitatively study woody cover, we had to classify it. And there are several approaches of classifying vegetation, but in terms of woody cover, the most widely used classification is the one provided by White, which takes into account the vegetation height, as well as the percentage of the land that's covered by woody vegetation. So it ranges from forests, which cover 80% of the land, greater than 80% of the landscape, and have trees taller than 10 meters, to grasslands, where the woody cover is less than 10% of the landscape. So moving forward, I'll be adopting this classification. So in terms of previous work on this seminal study by Serling et al. In 2011 was the first study to quantitatively estimate the fraction or the percentage of woody cover from the carbon isotopic signature of soil organic matter. And this study provided a model for predicting, or a model for reconstructing a woody cover from hominin sites. However, some critics have pointed out some possible limitations of this model because the study used a global, and the criticism is that it used a global data set and a soil sample set that are not representative of African environments. And this is for a few reasons. Therefore, this is where my work comes in. And here I'm trying to account for these issues and refine this woody cover estimation model using representative African ecosystems. So in the field, I used White's vegetation classification to identify different woody cover or vegetation types. And surface soil samples were collected using procedures that were previously outlined. So samples were collected along a transect in a 100-meter distance, and at each sampling location, because there's going to be differences in samples collected closer to a tree versus away from the tree, I used this approach where canopy versus gap samples were collected at each sampling location. And to estimate the fraction of woody cover at each sampling sites, I used Google Earth images at 100-meter square area, and that is going to account for how much of woody vegetation is in my specific sampling site. And once the soil samples are brought back to the lab, I conducted a soil texture analysis using the texture bifield method, where the soil texture test is performed based on the grit and clay content while the sample is wet. And for the isotope analysis, fine root samples from this fine root and plant remains in the soil were removed or were picked out. The samples were dried, weighed, homogenized, and the isotopic composition was measured using an isotope ratio mass spectrometer. So to show you the results, here we have soil textural differences across different woody vegetation types shown in this bubble plot on the left. And so in this plot, the bigger the circle, the larger the sample size for that specific soil textural class. And as you can see, the majority of the soil samples are sandy clay soils, with the next dominant soil type being sandy loam soils. And the figure on the right is basically showing the same information, but using the ternary USDA soil textural classes, showing the proportion of sand, silt, and clay. And again, as you can see, almost 70% of the sample is sandy clay soils, while about 14% are sandy loam soils. When we look at the correspondence analysis between soil texture and vegetation types, we see a strong association of sandy clay soils with relatively open environments such as grasslands, wooded grasslands, and bushlands. But we also see association of forests with sandy clay loam and sandy loam samples, which are relatively coarse-grained compared to sandy clay soils. And this is expected because grass-dominated habitats perform well on this fine textured soils, where the fine grass roots can penetrate and access water and resources at a much shallower depths. Whereas if you're in a tree-dominated habitat, such as a forest, more coarse-grained sediments are going to be abundant because, unlike grasses, they can access water from much deeper within the soil profile. So to show you the results for the carbon isotopic composition, so here this includes 152 samples, excluding some high altitude samples that might represent C3 grasses. So looking at the different woody cover types here, forests have a relatively narrow range in their carbon isotopic composition, with a median value of about -25. And most of the isotopic signatures are relatively narrow, except for the few samples in HSR, which represent gap samples. Woodlands, bushlands, and shrublands are combined here. They have a median value of around 80.4, and this group has a relatively a wider range in terms of the isotopic signal, because first it covers a wide range of woody cover. So in terms of percent woody cover, woodlands cover from about 80% to around 40%. But in terms of structure, they can range from closed, very closed woodlands, like what we see in Mago here, to relatively open bushlands, to what we see in Akagera. And wooded grasslands and grasslands have more or less similar values, with wooded grasslands having a median value of -13 and grasslands having a median value of -12.5. So when we look at the carbon isotopic composition of soil organic matter across this different woody cover types, and this is all the parks combined. So this box plot shows you that there is a trend across woody cover, and we also see statistically significant difference between forests and woodlands and bushlands. This is separated here, but also a statistically significant difference between bushlands, woodlands, shrublands, and wooded grasslands. But as we saw in the previous figure, we do not find a significant difference between wooded grasslands and grasslands. So using these results, an ordinary least square regression was conducted on transformed fraction of woody cover values. And here the best fit line is shown in blue with the 95% prediction interval. And based on this data set, we were able to provide a revised model of predicting woody cover or reconstructing woody cover. And to assess how the new model or the revised model performs on different data sets for the purpose of comparison here. So the figure on the left shows measured versus predicted fraction of woody cover values using the new model and using isotope data from the Serling et al. Paper, and it shows that the new model is over predicting fraction of woody cover. But if we use data from the study and the model provided by Serling et al., we see that there is under prediction of woody cover. And differences between the two models can be explained by relatively low delta certidity values for most vegetation types in the previous data set, but relatively higher values in this study. And there can be several reasons for that. Possible reasons can be if there has been a historical vegetation change in the area, if there are differences in the spatial scale that's used to calculate the fraction of woody cover, or it could be related to potential differences in soil texture, but the soil texture data in the Serling et al. Is not reported, so we cannot make comparisons here. But moving forward, since our data set is representative of Eastern African environments, we'll be using the revised model to make woody cover reconstructions. So now, applying our revised model to what we find in the fossil record, we used pedogenic carbonate data from the Omoterkana Basin that was compiled from the literature. And here, the probability distributions show the reconstructed fraction of woody cover. So in this figure, woody cover increases from the left to the right, as shown by the different shades in the legend. And just to compare the different models, the new and revised model is shown in dark green, and the older model is shown in light green. And if we focus on the Shangra Formation, we see that one, the new woody cover model is indicating a higher percentage of woody cover compared to the previous one. And two, we see that in the Shangra Formation, the earlier members B to G, had a higher percentage of woody cover, but after member G, we see this shift, or we see this decrease in woody cover. If we look at the other formations in Omoterkana, the general trend is similar, but we see that the Shangra is more wooded than the other two contemporaneous sites. So that concludes my work on modern ecosystems. And now moving to my work of studying the diet of herbivores, I will be looking at the diet of herbivores from the Shangra. And in terms of previous isotopic studies, there have been studies that looked at the diet of herbivores, but these studies, including a study by our own group, they focused on a few selected taxa to study the diet. So what we were trying to do in this study was to look at the collective herbivore assemblage and answer the question of, like, if we consider all the herbivores, what does the diet of these herbivores looks like, and how did it change through time? And this study is part of a collaborative work that we published last year. And for that, we used a large data set of more than 1,000 fossil herbivore dental specimens. And these specimens are housed at the National Museum of Ethiopia, where the sample collection took place. So we did powder enamel extractions at the museum, and the samples were brought back and analyzed at the University of South Florida. To quickly show you the results from this study. So we looked at 9 different mammalian families. As shown in the box plots here, the bovids, we analyzed around 5 different tribes of bovids, but here the data is presented at the family level. So for the bovids, we see a general trend towards increasing C4 resource consumption with time. So here the color shades, here's a legend for the color shades. So the more green indicates browsing, the yellow indicates grazing, and you can see the clear trend towards increased C4 grass consumption. And we see a statistically significant difference in the transition from member A to B and member B to C. And here, this is a guide to just show you what time is represented in these different members. In the cercopithecids, again, we see a trend towards increasing C4, and we see a significant difference in the transition from member B to member C. When we look at the dinosaurs, throughout the Shangra sequence, they had a pure C3 diet. When we look at the elephants, they start out as mixed feeders, but again, we see an increase towards C4 resource consumption. And again, we see a significant difference in the transition from member B to C. The same thing with equids. They were mostly C4 grazers, but this transition is noted by a significant shift. For the giraffes, we have a pure C3 signal throughout the sequence. Hippos show a mixed C3, C4 resource consumption with a higher percentage of C4 at times. For the rhinos, they fall mostly within the grazer range, except for a few outliers. And for the suids, we see this very clear trend towards increasing resource consumption. And also the significant change that we see in the other taxa is also observed in the transition from member B to C, and also from the transition from member G to H. So if we look at this data set in terms of the proportion of grazers, mixed feeders, and browsers, here, this ternary diagram is going to show you the proportion in each category. And ecologically, we see 3 different groups. First, we have this fauna, herbivore fossil fauna dominated by a higher proportion of mixed feeders, and that is for the earlier members of the formation. The second group is a mixed feeder grazer dominated group for the middle members, C, E, F, and G. And later we have a grazer dominated fauna in the upper members, H to L. And this corresponds with the statistically significant differences we observed that I just showed you in the previous slides. However, in order to compare the different members, since they do not represent equivalent temporal duration, we looked at the data at 200,000 year time beams. But again, we see a similar pattern. So prior to 2.9, we have this mixed feeder dominated group. Between 2.7 and 1.9, we have this mixed feeder grazer dominated group. But after 1.9, we see a higher proportion of grazers. We also conducted a non-parametric change point analysis to detect when does a major change or change in the distribution of the carbon isotope values occur. And combining all the 9 mammalian families here, we see that there is a significant change in distribution around 2.7, but also around 2 million years. And again, these change points are more or less congruent with the patterns we saw in the previous slides. But when we look at the different mammalian families individually, 5 taxa show major or significant change in the distribution of the delta 13C values. The bovids show 2 major change points at around 2.7 and at about 2.3. For the suids, there is a change at around 2.6 and at 2 million years. The elephants show a shift around 2.3. For cercopithecids, it's around 2.4. And for hippos, it's around 2.6. But overall, there might be a plus or minus, but again, this corresponds to what we already know in terms of the diet of herbivores. And earlier in our woody cover reconstructions, we saw that the Shangra was relatively more wooded compared to the other contemporaneous sites, such as the Kubifora and Nachukwi formations. And here we're looking at the diet of herbivores, and we're trying to compare that with these 2 different formations. So in these ternary plots, again, it's showing you the proportion of mixed feeders, browsers, and grazers. The different shapes indicate different temporal bins, and the colors are indicating different formations. And what's, again, interesting here is that the herbivore assemblage in the Shangra, prior to 2.5, it was mixed feeder dominated. Again, this is going to depend on the time being you use, but it becomes much more similar to that of Kubifora and Nachukwi only after 1.9 million years. So if you look at the herbivore assemblage in Nachukwi and Kubifora, after 2.5, they already have a higher percentage of grazers, but the Shangra matches that only after 1.9 million years. And this is interpreted as indicating the likely presence of this C3 dominated landscape along the Paleo-Omo River, which is usually considered as presenting a gallery forest refugium. So when we combine our vegetation reconstructions using the 2 different proxies that are used, that is using the woody cover reconstructions using pedogenic carbonates and using tooth enamel, despite showing a similar temporal trend, the vegetation reconstructions are not identical. And we saw that the woody cover reconstructions indicated more wooded vegetation setting prior to 2 million years, while the data from pedogenic carbonates indicated or showed that there is an increase in C4 grasses after 2.8 million years. And this plot is just to show you the disparity between the 2 proxies. And this is just a density plot, not a woody cover reconstruction, but a density plot of the raw isotope values converted to the carbon isotopic signature of the vegetation that it represents. And again, similarly, it represents that pedogenic carbonates is showing a more wooded component of a landscape, but the tooth enamel data is showing more C4 dominated signal. And this is likely representing the differences in the spatial-temporal scale represented by the two different proxies. And by that, I mean that it's likely that the pedogenic carbonates are recording the more consistent presence of woodlands along the floodplains. And since they are spatially restricted, they are giving us a much more limited signal of the vegetation of the landscape. But when we look at the diet from herbivores, herbivores generally seem to sample a larger landscape. So that means they are telling us about what's going on the larger landscape. And that is away from this floodplains or away from these fluvial depot centers. So in terms of summarizing the results, I mentioned that we see significant change in the diet of herbivores at around 2.7 and at about between 2 and 1.9 million years. So the question is, does this represent a real ecological signal? And the answer seems to be yes. And here, the results from the carbon isotope composition is shown along with other proxies. And in terms of this trends, or in terms of this temporal shifts, the major shift we see in the transition from a member B to C in the carbon isotope record is also matched by other proxies, such as the core distance, which is a measure of faunal dissimilarity or species dissimilarity across these members. But it's again also reflected in an increase in the proportion of light cover habitats inferred from the ecomorphological data. And similarly, the change we observe in the transition from member G to H between around 2 to 1.9 million years is matched by the pedogenic carbonates, where we see a shift after 2 million years. It's matched by the core distance, where the faunal assemblage is very different. It's again matched by the percentage of arid adapted taxa, which increases compared to the lower members. So we interpret this as this shift that we see between 2.9 and 2.7 is representing a real ecological signal in the Shangra. So to summarize what I presented to you, our revised woody cover reconstruction using the modern ecosystem work indicates a higher percentage of woody vegetation cover in the Shangra Formation from about 3.4 to around 2 million years. When we look at the diet of herbivores from the Shangra, we see an increase in C4 resource consumption across multiple taxa, indicating that more open landscapes or more open vegetation was becoming dominant with time. And when we combine the results from these two different proxies, we interpret this as indicating a consistent presence of gallery forests along this floodplains within a larger landscape that was becoming more open and dominated by C4 resources. And in terms of one of the major changes in the diet of herbivores, we see a significant change around 2.7 million years ago. When we tie that to the hominin fossil records, while the hominins from the Shangra that are older than 2.7 million years are attributed to Australopithecus, this shift that we observe around 2.7 coincides or is concurrent with first appearance of Paranthropus, the genus Paranthropus. And overall, this indicates that compared to Paranthropus and later Homo that occurred in the later record, Australopithecus was living in a more wooded or a C3 dominated landscape. But moving forward, if we combine this refined woody cover reconstructions from paleosols with other proxies that represent wider spatial scales like the Diet of Herbivores, we will have a more nuanced picture of what was going on the paleo landscape that was inhabited by early hominins. And with that, I thank my collaborators, different funding organizations and organizations that provide us with permits to conduct this works. And yeah, with that, I conclude.
Thank you so much, Enquye. Can you hear me now?
I can hear you.
Yes, there's a bit of delay between the YouTube signal and what we have here, but thank you so much. That was absolutely fascinating. Very, very, very thought-provoking talk with tremendous amount of data.
Thank you so much, Enquye.
That you have presented us with.
Hear me now?
I can hear you.
There's a bit of delay between the
Let me
YouTube
see. I
signal
think I
and,
have here.
what we have here, but
Okay, I'm back. And we'd like to take questions from people in the audience. Let me see if I can find the questions here. And yeah, I think people were writing some questions in the chat function in YouTube. So let me go to the first questions from our listeners. Do you think, Enquye, that the lack of difference between members B and C in the hippos could be because of their different niche, more water-centered niche, compared to many of the other taxa that you've looked at? So hippos, members B and C in relation to their environments.
Yes, it could be. But as I tried to show in the different taxa, there is a significant change from member B to C, but when we're looking at the change points, there are slight differences in the taxa that are considered. So it's likely that not every species is responding with the same pattern. There might be a bit of a lag in one taxa from the other. I can't hear you, Renee. Sorry. I can't hear you, Renee.
Now?
Oh, I can hear you now.
Okay. Let me go to another question from our audience then. There are modern ecosystems that have a long history of small-scale agricultural activities, for example, the Bossou forest in Guinea, even though many areas are now protected. would these be viable locations for modern vegetation studies like the ones that you're doing? And if so, is there a way that this can be taken into account and or corrected during your analyses?
That's actually a very good question. And yes, there are, even in the sites that I considered, there are a few parks that are impacted by humans compared to the others. So since that's going to highly determine the percent woody cover, it's, at least in my study, I tried to avoid these parks that have a higher impact of human disturbance, because we're introducing one aspect of introducing humans, and two, there is the aspect of introducing other herbivores which are domesticated. So I would say excluding those areas would be a wise thing to do, but there are also key sites that might be informative. So while working in the sites, one way to approach this is these parks, most of the parks are relatively, they cover a wide area. So if we're interested in studying these parks, we can try to minimize the impact by looking at parts of this national parks that are less affected by humans.
Mm-hmm. Very good. You can still hear me, I hope. Another question is, I mean, there's a very interesting, not discrepancy, but difference in, as you mentioned very clearly, between the data from the soils in the Shangra Formation. If we were just to look at the data from the soils, one would come away with a picture of a fairly wooded, very green, fairly lush environment. You mentioned gallery forest, riverine forest, but would this mean that you're sampling mostly near the axis of a big river? Is that where your samples are coming from for the Paleosol data? And maybe that's why you're getting such a consistently or significantly C3 signature. And somewhat related to that, is there an effect possibly of abundance data in terms of demography of more woodland or forest dwelling mammals compared to those who may live in the open plains, and that might also put the dental isotopic data towards the more open end of the spectrum? In other words, the question is about trying to get a bit deeper into resolving this, I wouldn't call it a discrepancy, but you have these two very rich sources of information telling us somewhat different things, which is not unexpected, but again, the issue of sampling locations and specimens numbers, does that play a role, you think?
Yes, really. Actually, you're right. Discrepancy is not maybe the best term to describe the two different records, but rather there is some difference. I think your first question is, are the paleosols sampling just closer to the actual river plain? And I think that's the case. And in terms of the Shangura, what we're interpreting is we have this extensive gallery forests that are compared to what we see in the lower portions of the basin that are extensive. So it's likely that the paleogenic carbonates are recording that part of the landscape. And in terms of the abundance question that you raised, I think that's very interesting. And we know that, for example, if we have this dense riverine forests and if we have primates within that, the fossil record is going to be limited because we're not going to have abundant preservation in that specific location. But in addition to that, you're right to bring this point, because even if we look at, for example, modern herbivorous assemblages. These grazers usually occur in a larger herd compared to what we find in the primates, right? So there is definitely one bias in like the actual ecosystem, but on top of that, there's going to be an issue of preservation when we're dealing with the fossil record. So it's going to be a combination of both. So yeah, we likely have that limitation with the herbivore record too.
Mm-hmm. Very good. Another question, Enquye, has to do with resolving these different environments, and the data that you show indicates that when you have a bushland, as in Mago Park, for example, isotopically that's different to distinguish from a woodland where you would have taller trees and very different species. So even though isotopically these, based on your data, they seem to be very close to each other, ecologically, it seems that they're very different vegetation categories, woodlands versus bushland. How do you deal with that when thinking about the past, when you cannot really see what the trees are or their taxonomic composition, just their isotopic signature?
Yes, thank you for asking this question, Renee, because this is one of the things that worries me when we're trying to reconstruct vegetation, especially in the hominin records, because there are these debates addressing that whether hominins were living in woodlands or whether they were living in wooded grasslands and bushlands. And like you mentioned, there is a huge difference ecologically speaking between woodlands and bushlands. In terms of the percent woody cover, they are basically the same. But if you look at the vertical vegetation structure, they're completely different. So woodlands have a much taller tree, and there is a wide range. They can be like very closed woodlands that resemble almost forests in some cases. But in bushlands, again, you might have open versus closed bushlands, but the trees are very shorter, the taxonomic composition is different, so they are very distinct. So one thing I would be cautious with my new or our new revised reconstructions is not to make this distinct classification, because we're dealing with a wide range. And in terms of how we can account for this in the future, I think we have to rely on other proxies that give us a signal of what type of taxa are there, but also maybe if, for example, using biomarkers, we can say something more about the type of vegetation. So the short answer would be to know the differences between these two distinct vegetation categories, we have to rely on other proxies.
And among these proxies, I know that for Omo, we have quite a bit of fossil wood, pollen, and yeah, at least for some of these members, there's fossil fruits as well. So this would certainly help with distinguishing different types of vegetation. But of course, pollen would phytoliths perhaps.
Yes, that's why we have to always go back to our multi-proxy approach, where we get the information one proxy cannot tell us with the other proxy.
And just a very specific question, you mentioned there Shungura, Cubifora, Nachukui. Does your data include anything from the Usno formation?
Yes, a little bit of, yeah, I should have actually mentioned that. Yeah, we have a little bit of data from the Usno, but the sample size is much smaller than what we have in the Shungura, because in the Usno, we're looking at the earlier members of member B.
And that's for teeth only, I imagine. No fossil soils.
No, this is the fossil soil data is compiled from the literature, but they looked at members B and above.
Yeah. Another question, Enquye, more on sort of some of the theoretical issues revolving around early hominins. You know the work of Elizabeth Verba, of course, that has inspired a lot of research. And Verba used the Omo Shungura data when she talked about the turnover pulse hypothesis. And although you're not looking at turnover pulses, you're looking at change points, which theoretically could coincide with a turnover of some kind. I guess some of that you've done with the core distance analyses, looking at different taxonomic compositions. But some of the previous work that you pointed out, some of the previous papers indicating shifts in the Omo region at 2.7 million years ago, or 2.8, and these would in part support indirectly this idea of some turnover taking place regionally and perhaps more on larger scales based on Verba's work at around that time. Now, some of your work also points to the time period around 2.7, 2.8, but you present a more complex picture of change points. What do you think about the turnover idea in relation to the data that you have, which again it's not the central focus of what you've looked at, but I think the implications of your work with the idea of a turnover pulse, more specifically in terms of Verba's work, 2.7 million years, would have something to contribute.
Okay, so in terms of the turnover pulse hypothesis, she was looking at the first and last appearance data of herbivores, right? So the hypothesis is that some herbivores become extinct after 2.8. This is slightly different from my work, which is only focused on the diet of herbivores. So I'm not looking at appearance and disappearance of taxa. I'm just looking at how does the diet of taxa change through time. And I cannot directly speak to that, but previous work from the entire Omoterkana Basin has shown that instead of this turnover pulse, there is actually gradual replacement of taxa. So I think that addresses whether there is a record of turnover pulse or not. And in terms of addressing your change point question, like I said, different taxa show slightly different change points, but we have to, again, take into account that while this is still a time series analysis, it's still biased by the fossil record. So we definitely see a change from member B to member C. But like if you look at the change point analysis, we see the change point after the first members of member C, around C4, unit C4. So not at the junction between the two members, but at the higher level. And this is mainly due to the lack of, if we look at the record between 2.9 and 2.7, the sample size is not that great for that specific time interval. So so I try to present this as a range, saying there is a significant ecological change between 2.9 to 2.7 rather than pinpointing, because in order to pinpoint a specific time frame, we need to have a fossil record that is equivalent, if that makes sense.
Yeah, very good. Another question here, Enquye. we often look at these diagrams that show isotope values, and then like you've shown, and then how these translate into a percentage of woody cover. Now, based on your work, we're going to have to shift all of our graphs that have been published in the past. True? Yes. Now, that's fine. That is absolutely fine. But the question here is, since we use this idea of percentage of woody cover as a way to help us think about hominin environments and about their adaptations to these environments, the fact that your work is going to make us shift basically all of these values towards more closed environments, correct? So in other words, for any particular place, for any particular range of values, you're showing here that that based on the new calibration, these environments would have been, as in Shungura, more wooded than what we have in previous reconstructions. What are the implications for this? What are the implications of this for understanding the place of hominins and their adaptations and their ecological roles in these Eastern African sites?
Okay, so in terms of changing our different figures, I think actually the work supports what previous studies have shown instead of changing it. But like you said, in terms of the woody cover reconstructions, it significantly shows a higher proportion of woodlands. And what that would mean for hominins is, again, since we're dealing with pedogenic carbonates that are spatially limited, what this means is that compared to what we taught earlier, more wooded habitats were available for hominins. But again, like I tried to show in comparison with the diet of herbivores, it does not necessarily mean that the entire landscape was wooded. It just means there were portions of the landscape that were more wooded that were available to hominids.
Very good.
And one thing that I would add in relating to your previous question is, again, we had this debates whether certain environment was wooded or not. And again, I would be very cautious in over-interpreting this woodland signal, because we might be dealing with relatively closer woodlands or bushlands, but at least using this data set, we're not able to make the distinction between the two. So I'd be very cautious in this interpretations.
Very good. Well, thank you, Enquye. I think we've taken quite a bit of your time, and we really appreciate this talk. Again, the topic is a very important one, both in terms of modern ecology, paleoecology, and paleoanthropology. So thank you so much for talking with us about this. And we've had, I think, enough questions for you today. So we will end our presentation now at this point. And just a reminder that next week, next Tuesday, we will have the second presentation of the series with Sergio Almecija talking about apes and human evolution. So another very, very interesting topic. I hope you will all join us for that discussion. And thank you so much, Enquye. We've really enjoyed this. Really appreciate you taking the time to speak with us. And we're going to go off now.
Thank you, Renee.